So let's start with the Omim. You have to just write on Google Omim. This page will come, which is Human Genetic Catalog, Online Mendelian Inheritance in Men. This page tells you each and everything about the gene of your study. Okay, so you have to write here gene like ALDH1A, which one, suppose this gene. Okay, write any gene of whatever is your gene. So when you will type, this page will come. This page will tell you about all information. It has many things, right? So whatever you have written here, the most matching one, open it. Okay, so there are many LDH1, there are many LDH. It means that it is so many families of this gene type, the LDH1A, H2, B1, and so on. So you should mention the exact match, like LDH1A1, because I didn't mention the exact name, so it's telling me like this. So this page is important. You can save it, copy paste it, or whatever. From this page number, you have to use information for your thesis, results, and discussion as well. So here the first thing is mention the full name of whatever the gene is. You can get aldehyde dehydrogenase 1 family, member A1, ALDAH1A. Okay, it has given alternate names. So yes, if somewhere else you will see the ALDH liver cytosolic retinal dehydrogenase, that is also the same gene with alternate names. Okay, then it mention you exact location. This is the gene symbol, this is the cytogenetic location on chromosome 9 Q21.13. Okay, genomic coordinates are like, these are the genome assembly. I told you earlier, if you forget, human genome, this is the human genome assembly 38. This is a recent one, means this is a reference human genome which is present in UCSC and NCBI and different sites in the world. This is a reference human genome assembly which is used as a reference to compare whatever you study. Okay, like for every organism there is a reference genome. For human, this is a recent genome assembly that we are using. Okay, for your gene, its location is on chromosome number 9, position number 72,000. It means 72 mega base pair from this position number 6712053. Okay, this is the location of your gene exactly. If you will click this, it will open the NCBI. Then comes the text, yani description of this gene. ALDH1 gene encodes what it codes. It tell you the disease. It encodes liver systolic isoform of acetaldehyde dehydrogenase, an enzyme which involved in the major pathway of the alcoholic metabolism after alcohol dehydrogenase. So liver metabolic disorder, it can be used as well. Variation in this has been implicated in different responses to the alcohol ingestion. So each and everything about your gene is mentioned here. Okay, you have to search on it. You should know what is written. Google it more. Okay, for everything their references are given, so you can open them and search more, or use AI to like simplify these words for you. If you are too, yeah, if you cannot understand this, take the AI to make it easy for you. Okay, although the geneticist should know these terminologies, if you know, please learn it. Okay, so what I mean by, if you are ready for your thesis write up, I'll ask particularly for your gene as well. Okay, you should know everything about your gene that you're gonna write in your thesis in the result and discussion session. Then comes the cloning result, that these et al, they have purified this gene from the human erythrocytes, and what are their findings they have mentioned. Okay, this determined that amino acid sequence of this is isoform monomer is composed of 500 residues and so on. This is the extra things that you can find here on the cloning and expression. This expression tells you that which organ, this gene is expressed. Okay, this is important for you. For the genetics mapping is important, like what is gene and who worked on it. So you can see for this gene in 1985-86, these two research papers are published, which Su et al. They have published it first. They have mapped the cDNA probes of by the southern blotting, by the somatic hybridization, and yeah, they have actually made it cDNA first time. Okay, this this author and its group. Then comes the gene structure. If you want to know the genes, what is the gene like? It is 53 kb long, divided into, it has 13 exon. Similar intron exon organization was found in ALDH21. They are mentioning that the gene family ALDH. Yes, you can mention this gene has two gene families, H1 and H2, which are quite similar in their gene organization. And we can call it as isozymes, means the same, because this particular gene is about the enzyme, so they have mentioned that isozyme, like that the genes are having the same forms. Then comes the gene function. If this gene is important, function is important because whatever the gene is functioning, that is affecting the phenotype of the your patients. So for this particular gene in 2015, came et al. They studied and they showed that the GABA correlates in the midbrain dopamine neurons. So you can see it's so much important for the neurological disorder. GABA pathway is involved in it. Midbrain dopamine neurons is going to affect. So of course this LDH1 is for sure, if there is any mutation, is going to affect the patient's neurological pathway. So this thing you can read this paper in detail, ask the AI, and search on the gene card, and you can discuss in your thesis if it is directly relating to the neurological, any of the neurological pathway, developmental delays, and so on. And then comes the molecular genetics. This molecular genetics, this last part, the molecular genetics, you can directly go to this one you must mention in your discussion, because here they have mentioned that in which year, who has published families for this gene. So they have mentioned that Thomas et al. They found low systolic LDH in the liver alcoholic patient with fatty liver mitochondrial. This was normal and shown with the low cortisol level. Okay, then they have mentioned in 1806 there is a Chinese family with a Chinese autopsy liver specimen. They have found it, and then they have done some experimental work. Then 1809 they have demonstrated that the Caucasian, what are the Caucasian? They are the, we call it to the Europeans, we don't say Europeans, in genetics we call it Caucasian. So this is was it means this family, this gene is also known in the European families, because they mentioned abnormalities in nine unrelated Caucasian alcoholic, nine different families. They have found this mutation and causing the syndrome. Okay, so this is how in this molecular genetics that things whatever is mentioned, they are directly related to your thesis, and you must mention in the thesis along with the references already given, the same references. Then this is a more advanced in the discussion, and you can mention this, that the animal model whatever is done on this study, who has done, let's say in 2007, Zhou Zinkon, they have done the animal modeling. If animal modeling is done on some gene, this is kind of a more, you can say, convincing data in terms of this gene is very widely understood. After that animal model can be made. Okay, it's not a new gene, so this is well studied. And animal model, if proves something, it means yes, this is the causative thing in human as well. So they have mentioned about the animal models. There may be some of the cellular studies, like they can do study on some of the cellular pathways and then mention here, and these references are given. Okay,